first question was to show how respiration functions in a bird lung and explain why partial pressure, dissolved oxygen, can be near 160 millimeters of mercury, close to the amount in the atmosphere, while the partial pressure in the blood of mammals only gets up to about 120 at most and usually isn't even that high. So here's a bird, here's the head and the eye, and showing the path for air. In the first part, in an inhalation, all of the air sacs, very important, all the air sacs expand. So these are getting bigger and these are getting bigger. And so air is pulled up into the posterior air sac, but air that was in the posterior air sac is pulled through the parabronchi and into the anterior air sac. So during inhalation, you have air moving through the parabronchi. The air is coming in at about 160, and so the air entering the parabronchi, the gas exchange region here, is at about 160. During exhalation, all of the air sacs compress, and during inhalation, this valve was closed, therefore the only path for air was like so. But during exhalation, this valve is closed. And so when all of these compress, that certainly pushes air from the posterior air sacs through the parabronchi, and the air that's in the anterior sacs is forced out. This is open right here. So air is forced out. So again, during exhalation, the green here, air is moved through the parabronchi. During inhalation, the purple, air is moved through the parabronchi. And during exhalation in green, air is moved through the parabronchi and out. If we zoom in on the parabronchi, we have air at 160 millimeters of mercury here, and the blood manages, it's not quite a perfect countercurrent exchange, so it doesn't quite get to this, but it does do much better than blood in a mammal. It can get up fairly close to that. And so the whole point is that the air capillaries and the one-way flow of air allows more oxygen into the blood. Second question was to diagram the path of oxygen from the atmosphere into the muscle cell of an insect, and then propose an argument for why insects might need less oxygen-binding proteins than a mammal. So here's the outside of an insect. There's the spiracle right here. And some insects have a tidal lung in which they can switch from being fairly compressed to bending part of the lung out, which will pull air in. But for many insects, it appears to be just passive. And the oxygen moves in. All of this is air. The very tips of these actually have fluid in them. And oxygen can pass into the tissues and be used up in cells, and then so the oxygen is moving into the tissues, being used in cells for respiration, in mitochondria, converted to carbon dioxide, and carbon dioxide diffuses out. Here's the fluid and during the final tips, but oxygen can move through. It's a pretty short distance, only 100 microns or so, so oxygen will diffuse through. So why might insects have hemocyanin as an oxygen-binding protein? This answer proposed maybe it helps oxygen gets to specific target locations. So that's certainly the case. Wherever you had some oxygen-binding proteins, you would have more oxygen moving to that location. It also provides a brief period of storage. It causes faster movement of O2 because because as the oxygen moves in, wherever there's a binding protein for it, that oxygen gets trapped. It can't go back, and so there's a net movement until the oxygen is bound. So multiple possible reasons. Next one was to draw an oxygen dissociation curve for hemoglobin. And humans. And make sure to label the axes, and then for human exercising heavily, what percentage of the oxygen in the blood is likely to be released, or you could also look at it as what proportion is still going to be bound to hemoglobin. So here is from 0 to 100% saturation of hemoglobin. And remember that in humans, it varies in different animals, but in humans we hold about 20 times more oxygen in the blood than we would if we didn't have hemoglobin. Different animals have different amounts of hemoglobin, so not all hold the same amount of oxygen. So we hold about 200 milliliters of oxygen per liter, but 95% of that is bound to hemoglobin. So in the lung, that would be up here. In the lung, we'd expect nearly 100% saturated. In muscle that's working very hard, that would be down here. It's using up almost all its oxygen, so it might be at only 5 or 10 millimeters of mercury down here. And so most of the oxygen at that point will have been lost. Okay. Muscle that's working but not quite as hard might be somewhere in here. If the heart is pumping hard, the person is exercising, that might be somewhere down around in here. 70% or so of the oxygen is being released. Whereas during exercise, not much is going to the stomach, so the stomach is not doing much, and not much oxygen is being released there. Most of the oxygen is still present. This was just another example showing these distributions. And the fourth question was an oxygen dissociation curve for myoglobin in humans. So myoglobin, so if we were looking at, this is 80 here and 160 here. If we were looking at hemoglobin, it would look like this, with near 100% at around 80-90 millimeters of mercury. But myoglobin is a very steep curve like this. It actually doesn't even have this little s. It's just a steep curve like this. So myoglobin isn't releasing much oxygen even when we're getting down around 30-40 millimeters of mercury. It's when the muscle is working very hard that it starts releasing oxygen. And here was another example. I would say that muscle working very hard is probably more in this region, and that's when some of the oxygen from myoglobin is being released to the muscle.